Welcome everyone, I'm Alicia.
And I'm Kay.
We're from ABARES and it's our pleasure to welcome you to the 2022 Science and Innovation Awards Showcase.
The Science and Innovation Awards for Young People in Agriculture, Fisheries and Forestry is an annual competitive grants program supporting young scientists, researchers and innovators. The program recognises original projects that aim to keep Australia's rural industries sustainable and profitable.
We'd like to thank our award partners for their ongoing support and commitment, and we're thrilled to have them introducing the winners today.
This year we awarded 12 grants, and very soon the recipients will give a short lightning talk about their award-winning projects.
Stay tuned right to the end when the Minister for Agriculture and Northern Australia, David Littleproud, will announce the winner of the Minister's Award. The award provides additional funding for one of our 12 category winners to extend their project.
So we'll now hear from each of our award partners, followed by each award winner's lightning talk.
Let's kick things off with our first award partner, AgriFutures Australia.
Hello everyone, my name is Laura Skipworth and I'm the Manager Emerging Industries at AgriFutures Australia. We're proud to introduce our winner, Dr. Ashwin Vadavalu from Murdoch University. We believe that Ashwin's project will give a great impact on the growth and development of the microalgae industry, as well as provide subsequent benefits to many emerging crops such as pomegranate and jackfruit. We are really excited to see the results as Ashwin undertakes his project over the next 12 months. Congratulations again, Ashwin.
A very good morning, everyone. Thank you very much for the wonderful introduction, Laura. I appreciate it. So basically, my project looks into the use of wastewater grown microalgae as a source of biofertilizer and also as a source of biostimulants for the cultivation of emerging crops such as jackfruit and pomegranate. The main aim of this waste to profit project is for the capture and conversion of waste nutrients such as nitrogen and phosphorus that are typically abundant in various agricultural wastewater into microalgal biomass, which can then be subsequently used as a biofertilizer for crop production. For anyone who's not very familiar with microalgae are nature's most efficient single-celled photosynthetic organism that are not only extremely versatile, but also extremely robust due to their ability to grow in a wide range of different environments, such as wastewater. Microalgae can also be an extremely valuable biofertilizer due to their fast growth rates and also the ability to release nutrients in a slow phase and also capturing the nutrients in a non-soluble form. They can also be an attractive biostimulant due to the fact that they can produce a wide range of different growth promoting metabolites such as phytohormones and polysaccharides, which can significantly improve the growth of various different crops. As such, through this particular project, I will be evaluating and optimizing the use of wastewater grown algal biomass as a biofertilizer for the cultivation of two different emerging crops, such as pomegranate and jackfruit. Also, as part of this project, I would be also comparing the efficiency of this produced algal biofertilizer to conventional alternatives that are currently in use, as well as study the effects of this biofertilizer to the fertility of soil. The outcome of this project is expected to equip and provide valuable information to the agricultural sector on the potential and also the advantages of wastewater grown algal biomass for the cultivation of various crops, as well as provide an alternative waste management strategy for wastewater treatment within the agricultural sector. So that's basically it from me for today. Before I go, I would like to sincerely thank AgriFutures Australia for their kindness in supporting and funding this project. Thank you all for listening and wish you all a great weekend ahead. Thank you.
Good morning all. I am Georgia Clark, the Project Manager for Innovation and Development over at Australian Eggs. We are thrilled to announce Dr. Amy Moss from the University of New England as our 2022 winner. Over the next 12 months, Amy will be taking a novel approach to precision nutrition for laying hens through the application of minerals through the drinking water, We are very much looking forward to seeing the impact of this research on layer health, egg production and the farm bottom line. It is a really novel approach that we haven't seen before. Congratulations does go to Amy, who will now share more of her project.
Thanks, Georgia. Hi, everyone. Like many Australians, eggs are an important part of my diet, and the demand for them is growing. On average, each one of us eats about 250 eggs a year. But with the rising cost of feed, producers are looking for ways to produce eggs more efficiently and sustainably. So precision layer nutrition has been of increasing interest. Precision nutrition is simply feeding an animal so that it can meet its daily nutrient requirements, preventing the waste of nutrients. Because of the daily cycle of laying an egg, layer hens are special in that they have cyclic nutrient requirements, where they need more energy and protein in the morning to form the egg yolk and the egg white, and they need more calcium in the evening when they're forming the egg shell. So hens usually benefit from having two diets throughout the day to meet these separate needs. But many farms only have one feed silo, so implementing two diets is impractical. Therefore, my project explores boosting the calcium level in the evening via the supplementation of liquid minerals to the drinking water, thereby achieving precision layer nutrition whilst maintaining the practical implementation for producers. This will allow the implementation of precision nutrition to the Australian egg industry, improving efficiency and sustainability. So a big thanks to Australian Eggs and ABARES for choosing my project.
Good morning, everyone. My name's Amanda Carter, and I'm the People and Culture Manager for the Australian Meat Processing Corporation. We're very excited and proud to introduce our winner, Sonia Moyes from Murdoch University. We believe Sonia's project on assessing the residual glycogen on sensory attributes of Australia lamb will have a positive impact across our industry. Once again, we'd like to thank Sonia and we look forward to monitoring and working with her on her project over the next 12 months.
Thank you very much for the introduction, Amanda. I would firstly like to say what a privilege it is to be receiving this award from AMPC and ABARES today. I am grateful for this opportunity and eager to see what this project leads to. So I'm currently undertaking my PhD looking at lamb eating quality, and we already know that eating quality drives consumer repeat purchasing behaviour, and predicting the final eating quality outcome is a concept uniquely used within Australia through the Meat Standards Australia system. Under the current Meat Standards Australia system for lamb, which is a best practice pathway system, there is still variation in eating quality, and this highlights the importance of understanding factors affecting palatability. So for this study, we are going to be looking at one of these factors called residual glycogen, and we will examine its potential impact on eating quality. So firstly, what is residual glycogen? Well, after animals are slaughtered, anaerobic metabolism in muscles converts glycogen to lactic acid, which lowers the muscle pH. So when sufficient glycogen concentrations are available at the time of slaughter, muscle pH declines to around 5.5, and then there's residual glycogen that remains in muscles. However, when glycogen levels are low at the time of slaughter, muscle pH remains high and there is no residual glycogen present. This leads to meat of reduced quality and decreased shelf life. Glycogen concentration is greatly impacted by an animal's nutritional status and pre-slaughter stress, and under the current Meat Standards Australia system, there are nutritional guidelines which can be implemented by producers to help achieve the recommended glycogen levels at slaughter. As we know that pH is an imprecise measure of muscle glycogen, within our project, we will be taking muscle samples to examine the impact of muscle glycogen on eating quality. This is novel research in understanding the relationship of this fundamental biochemical component and eating quality, and results of this study will be tested in a refined sheep meat Standards Australia model to test residual glycogen as a predictor of eating quality. Now, this may lead to glycogen threshold levels being included within the Meat Standards Australia model to calculate the expected eating quality of different lamb cuts. Our project is focusing on measuring these glycogen levels within one of the commercial high quality cuts, which is the loin, and we are hoping to expand this further and measure it on various cuts throughout the carcass to optimise the eating quality prediction for different cuts. Thank you.
Good morning. My name is Rebecca Athan and I'm the Manager for Production Innovation at Australian Pork Limited, and we are proud to introduce our winner, Lauren Stavely from Sunpork. Lauren's project is very exciting for us as it addresses one of our key pillars, our people. As our industry looks to navigate the changes around sow housing during lactation, our focus is not only on our pigs and their wellbeing, but also that of our stock people and how they adapt to and work within different systems. Lauren's project will go a long way to helping us achieve this and for both, the best outcomes for both our pigs and our people. Our industry is excited to see the results as Lauren undertakes her project over the next 12 months. So now I'll hand over to Lauren, who will give you a brief overview of her project. Congratulations again, Lauren.
Thanks for the introduction, Beck, and thank you to Australian Pork Limited and the Department of Agriculture, Water and Environment for supporting this project. So the Australian pork industry is proactive in its approach to animal welfare, proven by the voluntary ban on the use of sow stalls that confine the movement of sows during pregnancy. Sows are still confined in a piglet protection crate while they rear their litter, and this was designed for piglet safety, as it prevents the sow from lying down quickly and accidentally crushing her piglets. A lot of research has occurred to move the industry away from the confinement of sows during farrowing and lactation, and there are currently many free farrowing systems available. However, industry uptake has been slow. This is most likely as research from our group has shown that the relative risk of a piglet dying in a free farrowing system is 14% higher than in a crate. And while the design of these free farrowing systems is important, the role of the stock person is often overlooked. The attitude and skill level of piggery staff can have large impacts on improving the survivability of piglets. So to put it simply, staff that enjoy working in a particular system will make that system work. So my Innovation Award will involve a series of experiments that look specifically at staff that work in both farrowing crates and free farrowing systems. The experiments will involve surveying piggery staff to quantify their attitudes towards the two systems. Given that these are the people that work day in, day out with the sows and piglets, their opinion and observations should be quantified. And we'll also be employing a technique called qualitative behavioural assessment, or QBA, which is the human scoring of an animal's behavioural expression. So QBA relies on the experience of the stock person to interpret the demeanour of an animal and allows for the use of descriptors that have expressive associations to the animal. So, for example, an animal may be referred to as calm, anxious, excited, or agitated, which is reflective of how the animal is experiencing their environment, and therefore it's reflective of their state of welfare. So this project is truly novel in that it's the first to tackle the problem of reduced piglet survival in free farrowing systems through the role that staff play rather than modifications to design or sow piglet related factors. We're hopeful that the outcomes from this project can be used to help develop training aids and upskill staff working in these systems, as having the right staff will improve piglet survival and allow the pork industry to have more confidence in free farrowing systems. Thank you for listening.
Hi all, my name is George Lehman and I'm a project coordinator for training and education at Australian Wool Innovation. We are proud to introduce our winner, Bobby Lewis Bader from the University of Adelaide. We believe her project will have a positive impact on our industry and are really excited to see the results, especially as her study is the first of its kind to investigate the impact of hot weather in rams on farm rather than in climate-controlled rooms. We look forward to seeing Bobby undertake her project over the next 12 months. Congrats, Bobby.
Yeah, thank you, George. Firstly, I just want to say how grateful I am to have received the award and just how excited I am about getting the opportunity to expand my research, thanks to ABARES and the team at AWI. I believe my research truly supports this idea of growing agriculture through innovation and sustainability, especially throughout a changing climate. My industry award really complements my existing PhD research, which is focused on investigating the physiological and behavioural responses to heat stress in merino ewes. So I'm in the midst of running a trial this summer, which is primarily using sensory technology to remotely monitor core temperature in free-range systems. which I will then be linking to their movement patterns and resource use, reproductive output, as well as lamb growth and development, both in utero and postpartum. So since receiving the Science and Innovation Award, I have the opportunity to expand this research to investigate the effects of heat stress in rams as well, which is great because they are equally as, if not more important to manage if we want to reduce the impact of climate, current and future climates. The main aim of the project is to continue utilising innovative technology to investigate the importance of both scrotal and body temperature regulation during heat exposure, and the impact this may have on semen quality and fertility in merino rams. I'll also be introducing a behavioural component as well, so looking at movement and activity patterns under heat load, and how this might influence their following reproductive success. Additionally, the rams I will be using are going to be the male progeny from my current PhD trial, which is super exciting. It'll be the first of its kind to investigate the transgenerational patterns of thermotolerance. So does a ewe's temperature status influence ram-lamb thermotolerance? So I think this research will answer some important questions around how sheep cope with harsh ambient conditions in extensive grazing systems, and whether sensory technologies have the capacity to identify ewe and rams with improved heat resilience. The end goal, I guess, would be to provide producers with appropriate tools for efficient on-farm data collection, and a way that they could send the data off and receive recommendations on how to manage their flock during the summer months. So thank you.
Good morning, everyone. My name is Ian Taylor. I'm the Executive Director of the Cotton Research and Development Corporation. We are very pleased to introduce our winner, Dr. Xiaoqing Li from CSIRO Agriculture and Food. We believe that Xiaoqing's research project will enable full traceability of the Australian cotton throughout the supply chain, thus providing surety and provenance to our buyers. We are very excited to see the results as Xiaoqing undertakes her research project in the next 12 months. Congratulations, Xiaoqing.
Thank you, Ian, for the introduction. I'm Xiaoqing Li, and I'm a researcher at the CSIRO. Firstly, I want to say thank you to the CRDC and to ABRIS for choosing my project for the award and for the opportunity to give this talk. Our project on traceability has the potential to improve the transparency of fibers used in the textile industry. Traceability allows us to know the origin, quality, and the environmental and the social impact of cotton fibers. However, it is very challenging to track cotton accurately throughout the supply chain. This is because the textile supply chain is dispersed around the world, and the current technologies do not operate well across each stage. In particular, there is a lack of transparency in the middle processing steps. The current technologies for tracking cotton fibers have certain problems. Some approaches require special equipment and processing that is not easy to access, or require additional processing steps to attach materials on top of cotton fibers. Simplified methods rely on record keeping and sharing, which is not easily achieved with such a dispersed supply chain. In this award project, we will test a new concept to use plant-based genetic solutions to solve this industry issue. We will use various cotton germplasms developed in our CSIRO Synthetic Biology Future Science Platform project, which have unique property that we think could provide a traceable marker, even though it still looks like a normal cotton plant. With this approach, the goal is to eliminate the need for additional processing steps or complicated or costly equipment, reduce time needed to demonstrate source of cotton, and to reduce wastage. We hope this work can lead a new direction in developing plant-based tracing technology. If such technology can be developed, then we can accurately track Australian grown cotton. If successful, this will greatly improve the transparency of the whole textile supply chain and increase the value of Australian cotton. eventually to improve the sustainability of the whole textile industries. Thank you so much for listening to me, and thank you again, CRDC and ABERS, for this opportunity.
Hi, everyone. I'm Raghu Satyamurthy, the Research Director for Biosecurity at CSIRO. I'm excited to announce Dr. Thomas Schmidt from the University of Melbourne as the winner of the Biosecurity Digital Innovation Award. Tom's project combines his passion to combine digital and genomic tools to understand biosecurity threats. While the immediate focus of his project will be on a key group of insect pests of importance to horticulture, the approach he's taking is transferable to the management of other biosecurity threats. We're eagerly looking forward to the outputs of his research over the course of the next year, supported through this award, and any outcomes that may follow from that. Congratulations, Tom.
Thanks, Raghu. I'd like to thank my sponsors at CSIRO Biosecurity Digital Innovation to start with. So this project is going to be building a genomic data bank for a particular agricultural pest, and then using this to work out where invasions and incursions are coming from. So this is a genomic data bank is basically where you get a large number of individuals and you sequence their DNA, and then you can use that to insert patterns. So in this case, I'm going to be looking at a particular type of agricultural pest. I've worked on this sort of stuff with mosquitoes before, but this is a pest called Liriomyza trifolii, which is a leaf mining fly. And it causes all sorts of damage to all sorts of crops. So I won't really go into that here. The important thing is it's a global pest. It's all over. It's from the Americas originally. And it wasn't in Australia until last year when it showed up at 3 locations at the same time. Kununurra in WA, Katherine region in Northern Territory, and Cape York in Queensland. So this is 1,500 kilometres apart. Almost at the same time, this thing has just invaded all across the north of Australia. And we don't know where it's come from. And so that's a major problem to start with, because we don't know if it's coming from one location or multiple locations. But there's an additional bigger problem, which is that it's now going to start spreading into new locations. It's already been detected in Broome in WA, for instance, and it can come down into Eastern Australia through greenhouses. So this is the issue here is that we want to be able to work out not only where these invasions have come from, but when we find this thing showing up the first time, is this coming from other places in Australia that are already invaded or from somewhere else internationally? And the aim of this project will basically be once we've got DNA sequences from all over the world or major parts of the world, and then also these Australian invasions, we'll be able to trace all of these new incursions to where their original source is. And from that, we can build up ideas of risk around where it's coming from, which sort of pathways are most problematic. And then also we can extend this. This will be a sort of blueprint to be extending this into other species. This will be sort of the first deployment of this, to my knowledge, in an agricultural pest. But there's lots of advantages to developing it further. And one of the reasons I'll just end on saying why this is now coming into vogue is it's when I was doing my PhD research, 2016, I was paying $92 per gigabase. That's a unit of sort of sequencing. $92 per gigabase. Now it's around about $9 or $10. So it's reduced the cost reduction of 10 times cheaper. So it's really right now. It's all of a sudden we're able to do this sort of work. So this will be a launch pad for genomics and biosecurity, or genomics in biosecurity.
G'day, my name is Patrick Hone. I'm the Managing Director of the Fisheries R&D Corporation. And I'd just like to make a couple of comments. First of all, I'd like to thank ABARES for the opportunity today, for the opportunity to sponsor the Young Scientists Award. I'd like to congratulate all the winners. And particularly, I'd like to congratulate the FRDC award winner, Dr. Valentine Tipo. And I'd like to also make the comment that he comes from an amazing pedigree through Nick Paul's lab at the University of Sunshine Coast, and he's going to work with one of our favourite groups, which is Scott Parkinson and the Ornatus group and the Northern Australian CRC. So I will now pass it on to Valentine to talk about his project. Thank you.
Thank you, Patrick. So yes, my project will be investigating the potential of seaweed, the now famous red seaweed Asparagopsis taxiformis, as a functional ingredient in the diet of the Australian tiger prawn. I'm going to give you a bit of context and rationale why this project is both timely and quite important. First of all, functional ingredients are ingredients that are not providing the animal with key nutrients, but more promoting special traits in the animal. So I'll be mainly looking at the immune system of prawns and their productivity, so growth. In terms of the rationale behind this project, about 10% of the aquatic animals that we produce in aquaculture worldwide is lost to disease. So that's 1 out of 10. So that's equates to about $9 billion every year being lost. And it's also a lot of food that could be feeding a lot of people. Not only that, that's probably just the tip of the iceberg, unfortunately. The current way that we treat disease outbreak and prevent them from happening is through the use of veterinary drugs such as antibiotics. And antibiotics can lead to antibiotic-resistant bacteria, which are already claiming about millions of lives every year. And we have no cure for it. And that's projected to cost the global health system $1 trillion by 2050 annually. So we definitely need to do something about this. But what does seaweed has anything to do with it? Well, during my PhD, I found that feeding very small amounts of that red seaweed, Asparagopsis, to both rabbitfish and then later on Atlantic salmon, promoted their immune response by up to 400%. And a bigger and a stronger bolstered immune system also means the animals are more resistant to diseases, and it means that they don't require treatment with antibiotics. A cool side effect of that seaweed supplement was that the animal also grew faster by up to 19%. So what about crustaceans? They're very different from fish. So tiger prawns, very different from fish, yes, but they do have an immune system, and they're not immune to diseases either. Just 6 years ago, in 2016, we had an outbreak of a virus called the white syndrome virus, which was not present in Australia. And that wiped out many farms close to my home, actually, in central Queensland, in the Logan area. And again, we don't have a cure for that one either. So thanks to this project, to Avaris and our sponsors here at the FRDC, I'd like to provide prawn farmers, Australian prawn farmers, with a novel tool and an environmentally friendly tool to manage disease and maintain productivity, if not increase it. So that's me. And thank you, everyone. Thank you for all the winners. And thank you, Avaris, for organizing this amazing event. Cheers.
Hi, my name's Jodie Mason. I'm the Forest Research Manager at Forest and Wood Products Australia. We're proud to introduce our 2022 winner, Minkai Jong from Western Sydney University. We're really excited to see the results of Minkai undertakes his project, looking at the benefits and costs of soil amendments and impacts on carbon sequestration. We believe his project would have a positive impact on our industry and more broadly through optimising management for carbon dioxide removal by plantation forests. Congratulations, Minkai, and all the best for your project over the next 12 months.
Thank you, Jodie. It's a great honor to receive this award. And I want to thank Forest and Wood Products Australia and also the Department of Agriculture, Water and the Environment for funding this project. So I'm a researcher at Western Sydney University. In my background, you can see the world-leading facility UKFACE, where researchers have been dumping carbon dioxide into the forest to study its response to climate change. For this industry-awarded project, what I'm going to do is to study the soil amendment to increase carbon sequestration in the plantation industry. Wood production is a major commercial use of Australia's plantation forests. And also forest-based carbon credit is a very exciting new opportunity to co-benefit the industry and also the global effort to mitigate climate change. And this project was inspired by a recent global modeling paper where soil amendment using powdered basalt has been suggested as an economically viable and nature-based solution to increase ecosystem carbon storage. The primary mechanism is that enhanced weathering of basalt rock can release nutrients to alleviate nutrient deprivation in most Australian soils. There has been some evidence observed in cropland, but not much being observed in plantation industry. So this project aims to provide direct evidence on that benefit of this approach is that it can be co-deployed with existing land use. So potentially it's a one stone two birds scenario. What it does is that it can potentially increase biomass sequestration in the timber industry, and as well as provide extra carbon to offset global rise in atmospheric CO2. So potentially serving as a valuable carbon credit in the future. The outcome of the project will lead to an improved understanding of the costs and benefits of soil amendment in achieving the climate change mitigation target. And it will also identify potential pathways for considering existing plantation land for forest-based carbon credit in the future. Thank you.
My name is Ace Lawrence, Manager Soils Agronomy and Farming Systems at the Grains Research Development Corporation. We're proud to introduce our winner, Jaco Zandberg from the University of Western Australia. Jaco's project, entitled Silencing the Frost Damage Caused by Ice Nucleating Bacteria in Australian Fields, aims to ultimately assist grain growers in better managing frost risk through the use of a non-pathogenic, non-toxic, and non-GMO bacterial-based technology. Frost damage to wheat alone amounts on average to $360 million a year, and management strategies to deal with this abiotic stress are vital. We're really excited to see the results as Jaco undertakes this project over the next year. So congratulations again, Jaco.
Hello, everyone, and thank you. I'd like to first start off by thanking the GRDC and the Bears for this great opportunity and for sponsoring my project and myself. And secondly, I would like to give a small, brief description of my project and the technology that I'll be developing for the GRDC. So the project in question will be looking at tackling the severe damage caused by frost events here in Australian farmland. An excellent example will be in 2016, there was more than $1 billion worth of damage due to frost alone. And these frost events are not only severe, they are also reoccurring, with last year dealing severe damage as well. So the frost events themselves can be influenced particularly by an organism known as Pseudomonas. And in certain cases, this bacterial organism can contain a protein called an ice nucleating protein. And what that protein does effectively is it raises the temperature at which water freezes, meaning that the freezing happens earlier in the field, and also thawing is delayed, resulting in a greater loss in crops. Now, the technology in question will be targeting that specific protein and trying to suppress the activity of that protein. And the way we'll be doing that is by using a RNA-based interference molecule packaged into a naturally forming, non-toxic, non-GM or biodegradable nanostructure. And these are called outer membrane vesicles. And so when these interference molecules are packaged into these vesicles, they can be stored. And so an excellent thing about this project is that frost events only happen during wintertime. And so this product effectively can be stored and bulked. And so when the frost event is predicted by meteorologists, it can be applied directly to the field by the farmers. And so once applied, these vesicles will bud into the bacteria and effectively silencing the activity of those ice nucleating proteins, reducing the damage caused by alleviating some of the damage caused by the frost. And that's it. Thank you very much.
Hi everyone, my name is Bianca Cairns and I'm the R&D Manager for Training and Leadership at Hort Innovation, the horticulture industry's R&D corporation. I'm immensely proud today to introduce our 2022 winner, Dr. Stephanie Kerr from Queensland University of Technology. Hort Innovation believes Stephanie's project will have a transformational impact on the horticulture industry's ability to develop elite cultivars using new molecular breeding techniques. And we look forward to seeing the results as Stephanie undertakes her project over the next 12 months. Congratulations, Stephanie.
Thanks, Bianca, for the introduction. And thanks also to Hort Innovation for this exciting opportunity to look at different ways of engineering elite tree crop cultivars. So in order to develop elite cultivars with traits such as increased yield or disease resistance, it really helps to understand which genes are important in controlling these processes, as we can then target these genes in breeding programs. However, after starting in my current job a few years ago, where I've been working with tree crops such as macadamia and mango, I've come to realize that a lot of the molecular tools that we use in crops to understand gene function often don't work in these trees, which makes it really difficult to advance our knowledge about these species and really slows down the development of elite cultivars. So for my project, I want to develop genetic manipulation techniques that will actually work in these trees, and I'll be testing two novel methods in both macadamia and mango. So the first method that I'll be looking at uses nanoparticles to deliver molecules into the trees themselves to silence genes, so we can then look at what happens when these genes are no longer expressed. So this technique is really exciting, as nanoparticles are so small that they should be able to easily penetrate the plant cell wall, which is one of the major barriers for current techniques not working. The second technique uses a more blunt force approach to overcome the plant cell wall by using a microdermal needle to puncture tiny holes, which should then allow for the uptake of a bacteria called Agrobacterium tumefaciens, which is commonly used in plants to deliver molecules to affect gene function. So for my project, I'll be developing these two techniques in both macadamia and mango, and will hopefully allow for exciting advances to be made in both these species and aid in the future development of elite cultivars. So thanks again to Hort Innovation and for everyone for listening today.
Hi everyone, my name is Jo Hargreaves, Senior Program Manager at Wine Australia. We're thrilled to introduce our winner, Samantha Sawyer from the University of Tasmania. SAM's project aims to contribute to improvements in the way the wine industry makes decisions following the impact of fires and dealing with smoke-affected wine grapes. This will have a huge impact for our industry, and we're really excited to support Sam and see the results as she undertakes her project over the next 12 months. So congratulations to all of the winners, and congratulations again, Sam.
Thanks, Jo, and thanks for that introduction. So smoke-affected fruit is a significant cost to the wine industry, with potentially devastating consequences for those lucky enough to escape the bushfires themselves. This was most recently felt in 2019 and 2020. While the national loss was relatively low at 3%, bushfires and smoke taint impacted 18 wine regions, and maximum losses for individual growers were considerable. Climate change will only continue to exacerbate the issue. The impacts of smoke-affected fruit can be felt from vineyard owners through to winemakers, distributors, and retailers. It can impact the reputation of a whole region. The negative sensory aspects of smoke-affected fruit does not become apparent until after fermentation and aging, long after investments into the fruit and wine have been made with little to no chance for a financial return. Conventional smoke detectors are inadequate as they don't measure the concentrations of damaging smoke taint phenols, and lab-based analyses are expensive and time-consuming. What if we could create an early warning system of high smoke taint levels on a vineyard or from grapes in a winery to help them make decisions earlier based on their individual risk? That is how I will use this Science and Innovation Award. I'll explore two different approaches to develop proof of principle prototypes to measure smoke taint phenols, one based on colorimetric assays and the other on resistance-based sensors. The goal is to develop an affordable, simple to use, on-site smoke detection and measurement system to assist growers and winemakers to make decisions about a stop-go point for investment in fruit and processing. As researchers and scientists, I believe we should pursue both to expand the body of knowledge and to consider its use, to develop new technologies hand in hand with the people who could benefit so it has meaningful impact on the community sooner rather than later. I'm incredibly grateful for this award and would like to thank both Wine Australia and ABARES for giving me this opportunity to work with Wine Australia and their levy payers. If this project works, we can use this technology to help people sleep at night and worry less about the future of their passions, their businesses, and their livelihoods. Thank you for listening, and I'd like to congratulate all the winners as well.
Well, a big thank you to all of our award partners and congratulations to the winners. So now we're going to move on to Minister David Littleproud for the big announcement.
Well, great to be with you, albeit in a virtual sense. I'm pleased to announce that this year's Minister's Science and Innovation Award goes to a Queenslander, Dr. Stephanie Kerr, for her work on molecular speeding up of macadamia breeding in the industry up here in Queensland. Congratulations, as a proud Queenslander, couldn't be prouder that we won it again.
Thank you. I wasn't expecting to win the Minister's Award, so I'm not sure what to say. But yeah, I'm really honoured to be awarded both the Horn Innovation and the Minister's Award today.
Well, congratulations, Stephanie, and congratulations to all of our winners today. And thank you to our award partners and all of you joining us online for today's showcase. If you are interested in learning any more about our winners' projects, you can reach out to them into the chat. Or if you are interested in learning about Science and Innovation Awards, please visit our website, awe.gov.au/science awards. Or you can speak to the ABARES team in the Spotlight exhibition. But we will see you at 1:30 for the next session. Bye. Bye.